A real fundamental discussion about protein folding. And you guys, by now, you had General Biology 1, and you should have learned the primary structure is how the N-terminus, the primary structure is the amino acid sequence as you would write it out on a piece of paper, right? Primary structure of protein. So we know that all proteins in humans begin with which amino acid? Methionine, right? All proteins in humans begin with methionine. It's ATG, right? Which is the first codon for every gene, right? Yes?
Except for some exceptions. Don't worry about them right now.
All right, so we got methionine. And then we have what we call single letter codes. So that'd be alanine. This is glycine, aspartate, glutamate, leucine, tyrosine, lysine, I'm just making some up, histidine, I'm making some up, you can go on down, until you get to the final amino acid, and then you have your C-terminus. This is your N-terminus, right? The secondary structure is how the amino acids sort of interact with each other. And to remind you, this should be, maybe the amino acid kind of chain kind of coils around like this, and you get hydrogen bonds that form in here that kind of hold the structure stable, and that is called an alpha helix, right? So then you can have all these different structures. There's a beta sheet, there's another alpha helix, there's another alpha helix, and they're all out here. That's the secondary structure. The tertiary structure, the final fold of the protein, is how these things, if this is an alpha helix and this is an alpha helix, how they all kind of fold up and glom onto each other and interact to make the final three-dimensional shape of the protein. That's the tertiary structure, right? So what I've drawn here is specifically flawed. And generally, pardon me, I think I've got some kind of a cry going on here. Sorry about that. But generally, if the N-terminus is here, it's in what they call a random structure, or an unordered structure. There's different terms people apply to it. So this alpha helix is pretty stable. It's not like it's an alpha helix and then it like unfolds into a non-alpha helix and folds back up. That does happen to some small degree. They call it protein breathing. But in general, if my body is the alpha helix and this is the N-terminal part of the protein, and this is folded up into some secondary structure, the N-terminus never folds into some shape. And it's just kind of flapping around out here like this. Literally, as water molecules slam into it, it'll knock that thing around. Okay? So it's an unordered or available or loosely attached or something like this. And the same is with the C-terminus. They're unordered. So we were talking about, last time, and I drew like a little birthday cake. You know why that's in my mind? Today's my daughter's birthday. So I drew histones. So H2A, H2B, H3, and H4. And to get two of these, and two of these, and two of these, and two of these, they assemble into your what? Histone octamer. And if you take DNA and you wrap DNA twice around your histone octamer, you got your linker region, and then the DNA wraps around, it comes around, and wraps around, and then you have another linker region. That is called a what?
Nucleosome.
A nucleosome. I'm going to make this even more birthday cake-like. So the N-terminal portion of the histones and the C-terminal portion of the histones is not in this ordered shape of a nucleosome. The N-terminus of those 8 proteins is poking out. Like this. They're kind of like funky candles, okay? Like that. And we call these the N-terminal tails. So I'm loaded up here. daughter turned 4 today. It was very exciting at 6:30 in the morning when she had her first moment of like groggy wake up and like, it's my birthday! Running out, and Santa came. The house was messy with toys and crap all over the place. And when she woke up and ran out, the house was absolutely spotless. There were streamers everywhere, balloons hanging everywhere. The table was full of presents. The only thing she could think of is, Santa was here! We're going to have a Christmas tree, because I'm a Grinch. I don't like Christmas trees. Every morning when they wake up on Christmas morning, there's a tree and all kinds of presents under it, because Santa is unhappy that Daddy's a Grinch and brings a tree for the family. That's how it works. Thwarted again, that's awful Santa. I'm going to get him someday. Okay. So we have these N-terminal tails poking out here. And now I forgot where I was going with this. Okay, so we've talked about these linker regions here, right? This is where we ended last time. We talked about the DNA, the fragmentation assay here, where they exposed to restriction enzymes. And they showed that in between the linker regions gets cut, right? We showed that last time. Okay. So. Pardon me. Okay. I wasn't as bad this morning. So what happens is histone H1 comes in, and histone H1 sits on top of here like this. And it kind of stabilizes that structure. This is an electron micrograph image. So basically they have purified DNA that's associated, so they purified chromatin. The word chromatin means DNA plus proteins associated. Okay? They purified DNA, and they, for our purposes, we're going to say they dropped it onto a glass slide, and they blasted electrons through it. We haven't talked about how you make an electron micrograph, have we? Should I do that real quick? You saw it in the body. Actually, yeah, I was there. Okay, so you guys know how a light microscope works. Oh god, that's terrible. You think this one gonna work? Nope. Whoa, 2 for 2, I could play for the Nuggets. So let's say we have a glass slide. We have some kind of a sample. We want to see what's inside Dr. Ogg's fingers. So we take a knife and we go zap, cut off the finger, and we take a very, very thin section of the stump off there, and we take that and we set it on here. Incredibly thin, so thin that it's kind of translucent. We take the rest of my finger, stick it back on, and amazingly I heal up and I still have a finger, we'll say. No harm, no professors were harmed in the making of this micrograph. Okay, light shines up. So these are photons, which we all know are particles, right? So if you have an atom with electrons around it, and there's a nucleus with protons and neutrons in it, because these are particles, the particles, every now and then a particle would hit this and get absorbed and then emitted off in some direction, or I guess easier we can just say it gets knocked off in a different trajectory. Okay? So if I have a piece of photographic film up here, if light passes through, light will hit the film and turn that into a dark spot on the film, right? Is that correct? Or is it a bright spot? A bright spot. It will develop the film. Bright, yes. If there's something really dense in this spot, so that most of the photons are hitting this and getting zapped off in other directions, almost no light will get through and it will remain undeveloped. It will remain a dark spot on the film. Right? Get it? That makes sense? Same concept with an electron microscope. Instead of shooting photon particles, you're sending electron particles through. Right? If an electron hits this, it'll shear off and it'll not hit the detector up on top. Same concept, basically the same thing, right? So what they've done is they have a glass slide, they put DNA on it. In places where there's DNA, there are atoms. And so electrons going through there get deflected off, and the film does not develop. Areas where there's no DNA, those electrons will pass through those areas, and you develop the film up here. So electron micrographs are black or white. If you ever see the introductory textbook that we use for this campus, Brooker Biology, Life Science, or whatever it is, they have colored electron micrographs. It drives me insane. You can't have it. There's no such thing. Some artist went in with a pen afterwards and did that. Anyway, what you can actually see is the dark spots, and I have to turn the lights off so you guys can see this. Sorry for the camera here. You can actually see this is the line, the ribbon of the DNA, and each of these little dots here is a nucleosome. The DNA goes and then wraps 2 times around the histone octamer. The first time that this was identified, someone described that as like a bead necklace, the beads on a string. Okay? So it was pretty interesting. They could then add different chemicals. They could have purified DNA, and they could add different chemicals, and certain chemicals cause the DNA to compact, and for these beads, it can get very, very tightly associated with each other like this. And then other chemicals, maybe you purify some protein out of a cell, you dump that protein on here, and now this stuff opens up into this much more loose structure here. So the lights are coming back on. In case the camera couldn't see me while I was doing this, there's a protein you can add that will cause a loose DNA to compact like this. There's a different protein you can add that you can decompact DNA into something like this. So, we know that DNA wraps twice around a nucleosome. There are 3 levels of DNA packaging beyond that. I can't go into the details why. So you say, okay, the first one is wrapping DNA around, and then sensibly, the next one is how the histones interact with each other. And this has been proposed as a model that histone H1s kind of bring everything together. Look how compact the DNA is there. Doesn't that look awesome? And if you look at it, there's actually a spiral that runs around like this. You see that? It's actually a helical structure. Mind-blowing. If you get really high quality electron micrographic images, you see a spiral structure to the DNA. This has definitely got to be it. Here, someone else has proposed this model. It doesn't compact as well, but you can actually still kind of get kind of a spiral going on with this thing. So when I was in graduate school, everyone was like, this is it. This has got to be it. This is the way you pack it most densely. This is the most sensible way of doing it. There's experimental evidence that suggests this can happen, but more evidence suggests that this is probably the way it works. But we don't know. So, sorry. When I was in graduate school, we didn't know. Someone may have figured it out by now. If that has happened, I'm unaware of that because apparently I'm outdated. So, this is one of those wonderful moments. You learn how to add, you learn how to subtract, you learn how to multiply, divide, and exponents, all this stuff. You've been building your knowledge. You are now at the end of at least my knowledge. You're almost at the end of knowledge of how DNA structure exists. Isn't that cool? You're there. It's been 20 years. You're finally there. Isn't that cool? There are ways of compacting the DNA beyond this. And the goal that we talked about last time is DNA, if you took all the DNA out of one of my cells and lined it up end to end, it's about 6 feet tall. We need to compact that down smaller and smaller until it's small enough that it would fit in the nucleus. So wrapping around histones got us down to about 1 foot. The next level of compaction gets us down to maybe 1 inch. There needs to be 2 additional, and there's evidence for 2 additional ways of layers of compacting the DNA. So, if we look at the nucleus, here's the nuclear envelope going around like this, and the DNA is all in there. Turns out, if we were to examine this with a really, really high power microscope, so here's the inner nuclear membrane, here's Here's the outer nuclear membrane. Remember, the nucleus has two lipid membranes around it. Let's put some nuclear pores on it, like this, so things can go in and out of the DNA, or out of the nucleus here. And the DNA is kind of in here. And when I was in your shoes, we learned that DNA is randomly in there. DNA, after division, it just kind of uncoils itself, decompacts itself from the mitosis stuff, and then it just kind of flops itself out. Right? Like one of my college roommates would come home, flop himself on the couch, different every time. If we look at this, there's actually protein fibers. First of all, there's a protein layer that runs all underneath the surface. Those are called nuclear lamins. And there are also proteins that assemble into long, thin fibers, and they kind of penetrate through the cell like this, through the nucleus like this. And there's potential, well, I'm going to say there's potential here in this class, I guess later on, I'll say definitely. The DNA appears to interact with these filamentous kind of structures that form in the nucleoplasm. Okay. We know almost nothing about this internal structure of the nucleus. If you are interested in getting a PhD in an undiscovered area of biology, that's it. My guess is someone will win the Nobel Prize doing this research. And it's wide open. So it turns out, in our DNA, so this is going to be that kind of spinny curl that we talked about, the 30 nanometer fiber, where the histones all come together. I've proposed the two models. So that's this thing, it's that coil thing. So the DNA has been wrapped around the nucleosomes, and then the nucleosomes have interacted with each other to form this thing. And now those get looped together. And it would be basically like me taking this fiber here, this line, and going like this. Right? And tying all that together. And we saw this structure in bacteria, didn't we? We called this a rosette, right? We called this a rosette, the whole thing. And these are the radial loops of the rosette, radial loop of a rosette. And there just needs to be some central point that they can all connect to. And it appears that central point is more than just a spot in three-dimensional space. It's a filament. And you get a ring of radial loops here, and a ring of radial loops here. You get a rosette here and a rosette here, such that the row after row after row. This is likely these things. And there are consensus sequences. You know what a consensus sequence is now. There's a sequence which is found in the human genome, which is highly conserved, like the ALU sequence. The sequence of that DNA appears to bind to these proteins. So I'm not sure if the ALU sequence actually is the sequence. Remember we said the ALU is that 350 base pair thing that appears a million times in our genome. There are other things, there are other sequences that appear often in our genome. Those things bind to this. There's a reason we have them, not just that they're selfish. They perform a function of compacting the DNA like this. So those sequences are known. When I was in graduate school, they were not known. We knew that there was a connection point. We didn't know what the actual DNA sequence was. Okay? So we call those SAR. Scaffold attachment region. And I don't particularly care It's not like that is the answer to cancer and you need to know these things. We're going to come back to that a different way. It was an amazing thing that happened while I was in graduate school. Okay. Is there any questions on this level of DNA compaction? Packaging of DNA. You guys look like you're ready for a commercial break. You ready for a commercial break? So, when I started graduate school, I didn't know what laboratory I wanted to work in. And I was required to interview every single person in my program to learn about the research, to see if maybe I'd be interested in the research. So I interviewed these, and it was a husband-wife team, and they were studying how DNA compacts, how DNA wraps around histones, and how histones interact with each other. And they found that if they compacted DNA a certain way, the gene would be turned on. If they compacted DNA using different sets of proteins, the gene was going to be turned off. How the DNA got compacted seemed to control whether the gene was expressed or not. I was like, oh my gosh, this is incredible. The classic thinking of transcription is RNA polymerase binds, and then you make your RNA, your messenger RNA off that. If RNA polymerase can't bind, you don't get your messenger RNA. That's it. This is a whole new way of regulating gene expression. This is crazy. This is incredible. So I was really excited about it. And my brother is in the same area, but he's about 7 years farther along in his career than me. So he had gotten his PhD and he was doing his postdoc with me at the time. And I called him and I was talking about it. I said, this is incredibly exciting. I want to study histone DNA interactions because I think that's going to be a whole new area of gene regulation that nobody knows anything about. His advice to me was, nobody wants to study, nothing important comes from studying structural proteins. People are studying signaling pathways because that's the cause of cancer. You want to study how DNA wraps around a protein. It's a structural thing. It doesn't perform any important functions. Go work in a cell cycle lab. Go work in a cancer biology lab. Go work in a lab where they're studying how cells interact with each other, signal with each other, and sense the environment, because that's where the exciting research happens. While I was in graduate school, and so I heeded my brother's advice, and I did not choose this laboratory to work in. While I was in graduate school, it became absolutely clear that the way you package DNA regulates gene expression. And this packaging process is probably more important than the RNA polymerase making an RNA stuff you learned about. This is probably the most important way that genes are regulated. And we haven't even gotten into that yet in this class. And has a Nobel Prize been awarded for this yet or not? It's going to be. Someone's going to win the Nobel Prize in this area. But I studied cancer biology. Interesting commercial break.
It's kind of sad.
Kind of sad. Yeah. My brother's right more often than not. He's good. He's right. Okay. It turns out when you think of the shape of a chromosome, this is a chromosome that's been replicated, right? And that's in mitosis and it's going to separate, right? It turns out at the end of mitosis, you know, the DNA unravels and it kind of goes back into solution, right? If you go to a cell that is in S phase, and it's replicated its genome, but it hasn't gone through mitosis yet, so the DNA is all open, but it hasn't compacted yet. You can actually look, and this is the DNA, and this is protein that's been stained. This is this protein scaffold stuff in here. Even in an interface cell, in G1, cell hasn't even replicated its genome. The DNA remains bound to a protein scaffold, which is very reminiscent of the shape that chromosome will take when it compacts during mitosis. So basically, you've got it on the scaffold, and when it decompacts after mitosis, it kind of unravels, but it doesn't it's not like you've got some chromosome over here that was over here, and now the DNA unravels so that it's diffuse all throughout the whole nucleoplasm. if it's tied to some protein scaffold in this area, those radial loops can only go out a certain distance away, right? They can only be so big. And that suggests that in our cells, every chromosome has a defined spot where it must be. It's not random. Well, how the hell do you show that? Anyone want to take a guess?
You tag one of the chromosomes with a radioactive tag or a fluorescent tag.
Okay.
See where it goes.
So let's tag chromosome number 3. How are we going to tag chromosome 3 with a dye? And only chromosome 3. Chromosome 4 doesn't tag.
Would you tag it during the metaphase when it's condensed?
Okay. How are we going to tag 3 and not 4? You're going to go in and you're going to staple a dye. No, just kind of joking. How do you make it specific? You go in with little instruments and put something on 3 during mitosis?
Usually got a primer that uses like radioactive nucleotides. So
Okay.
replicate that one.
So you've got a primer and your dye is connected to the primer. How do you get the primer on chromosome 3 and not chromosome 4 and 5 and 6 and 7?
You just find some sort of sequence that's uniquely in that chromosome.
You find a sequence on chromosome 3 that's not found on any other chromosome. How likely are you to find a sequence on chromosome 3 that's not found on chromosome 4 or 5 or 6?
That's how many nucleotides you want to look at.
Yeah, okay. Depends how big the primer is. How big does the primer have to be specific? We did this once in class over. Do you guys remember how big the primer has to be? Is it 18? 17? If a primer is 17 nucleotides long, if you randomly made a sequence 17 nucleotides long, the odds of that sequence that you just randomly created appearing in the human genome is probably going to appear about once. So if you design, you're exactly right. If I design a primer that's 40 base pairs long, and that 40 base pairs I know is found in chromosome 3, that does not appear anywhere else unless it's a repetitive DNA sequence that we chose. So let's choose a gene that's on chromosome 3. Because that gene isn't going to be found someplace else in the genome, right? And that's exactly how you do it. It's called chromosome painting. And the way it works is we're going to take primers with a blue dye attached to them. And those primers anneal to chromosome 1. We're going to take primers with a red dye on them, and the sequence of those primers can only bind to chromosome 2. So chromosome 1 is blue, chromosome 2 is red. What color should chromosome 3 be? Purple? Okay, chromosome 3 is purple. Chromosome 4 should be what color?
Green.
Green, okay. And then what? 5? So you get like fuchsia, you get like magenta, light purple, dark purple. And this chromosome painting, it's standard technology. And every chromosome is labeled its own color. So if we do this, we can actually, here they've labeled chromosomes 7 and 8. Sorry, I'm going to turn the light off again real quick here. Chromosome 7 has been labeled light blue, and chromosome 8 has been labeled, what color is that? Reddishy thing? Maroon, should we call it maroon? It turns out, if you take a living cell and you label the DNA, in a healthy human cell, chromosome 7 is pretty much in the same spot every time in every cell. There's some variability on that. I guess I'm going to retract that. But basically, if that cell divides, the two daughter cells will put their chromosomes in the same spots. The DNA is put, it's like stored in a shelf in the nucleus in the same spot every time. Is that incredible? Nothing in cells happens by random, I think. I think everything is organized a specific way. Because that's how it evolved to be, right? What do you guys think of this? Okay. Oops, that's not what I'm doing. This one. So we can actually look at DNA in another way. If we have a chromosome, parts of it are actually very highly compacted and other parts are not. I hope you learned this in Bio 1. Parts of a chromosome that are highly compacted are called heterochromatin. And parts of DNA that are out loose and float around are called euchromatin. So this is heterochromatin, this is euchromatin, this is heterochromatin, this is euchromatin, this is heterochromatin. Telomeres and centromeres are always heterochromatin, are always highly compacted. And to the best of my knowledge, they are never euchromatin. No asterisk this time. Some portions are highly compacted and some portions are euchromatin. This is not what a typical chromosome looks like. You'll have, like, this area will be highly compacted, and this area will be highly compacted. You'll have spots of high compaction and spots that are all wide open. How is this controlled? Now we finally get to the histone tails. Turns out, what does phosphorylation mean? It's what?
Adding a phosphate?
You put a phosphate on protein, usually is what we think of. What does that? A kinase puts a phosphate through it, a PO4. So it takes ATP, takes that third phosphate and sticks it onto usually a serine or a threonine, sometimes a tyrosine, or protein. So now you phosphorylated protein. So it turns out in the histone tails, and I don't know the sequence of them, but I've written something up here that is probably not far off. In the sequence of these tails, there's an unusually large number of serines that can be phosphorylated, and also lysines. And it turns out, if phosphorylation means putting a phosphate group on, does anyone know what this functional group is? Come on, chemists. It's an acetyl group, yeah. It's an acetyl group. So lysine actually carries a positive charge at physiological pHs. But if you covalently connect an acetyl group on here, it binds into this spot, such that this is no longer positively charged. In this case, let's put another one right here. I've now acetylated this histone on the N-terminal tail. Okay, this changes the three-dimensional shape of this protein, right? I now have these big things hanging off here. What else have I done to this protein?
Made it not charged.
I've made it not charged. What charges DNA?
It's negative.
Very highly negatively charged. Does this protein want to be hanging out with DNA anymore? Not as much. If I take these off and I put these positive charges on, those guys go away, the positive charges return. DNA gloms on and hangs on really tightly. So it turns out whether DNA is heterochromatin or euchromatin floating out there depends on the modifications that have been put on the N-terminal tails of the histones. And this is something that's called the histone code. Well, if I put a phosphate here, the DNA compacts. But if I put a phosphate here, the DNA compacts. If I put a phosphate here, the DNA loosens up. Well, sometimes phosphorylation does this, and sometimes phosphorylation does this, I don't know. The one thing I can say with almost perfect certainty is if the histone tails of histones become acetylated, the DNA can no longer interact tightly. And so the DNA opens up. That's the only general statement we can make. We'll talk about the histone code in a little more detail. It all gets really, oh, we're done here. Yeah, so it's actually intended to be today's. And I don't have that. I don't know if I put that. Has anyone got Blackboard open right now? Do I have this next one on
I
Blackboard?
don't think so.
Nuts. means I have to do it here. I get to do it here. It doesn't work in this room very well. It may be done for, let's see. Oh, I opened up a Blackboard. Anyone know any good jokes?
Two snowmen were eating a carrot cake, and one snowman says, "This cake tastes like boogers."
Two snowmen are eating carrot cake, and one says, "This carrot cake tastes like boogers." That's a good one. Which one was Olaf? So my daughter got a Frozen storybook, kids book, and it's totally very, very reduced story of the movie, right? So I'm paging through it, and it's told from the perspective of Olaf. So I was doing my best, and I'm not going to do it. My best, but I'm taught, the Olaf voice, and my wife was like, "That is really good." It's my inner snowman coming out. Introduction to genetics. I saw some of you at the showing, Friday night of spring break. The Friday night that spring break began, I saw some of you there. Did I see you there? You there? Did you see me in the front there with my kids?
No, you got there early. I
Yeah,
got
I
there
did.
too late and didn't have a seat.
Yeah, I was lucky I got there early and my kids could spread out. That was awesome. Why were so many people not on spring break at that point? That was just crazy. I was like, oh, there's gonna be like 20 people. It'll be like people, from Texas or people from China that can't go do everything lined up. But it was over in Brown. How many people were there?
So many.
Yeah, the room was packed and there was no more room to stand.
What?
For Frozen. Yeah, it was good. It was a good time. Free popcorn. My daughters were loving it. Add an item.
Yeah, they were expensive.
Yes.
Because it was spring break.
Yeah. And those bring some new stuff that a lot of people are together, so we good for right? And nobody was working that day too.
Yeah, I'm supposed to start.
Genetics. Lectures.
Grade not learned subjects. What is that?
We got good 4 seats.
We're up in Florida. What does it mean? How did they know?
So Frozen came out on video.
Yeah, yes.
I'm infatuated with this movie apparently. My wife puts on the music soundtrack every morning, so I've got those songs running in my head.
Every morning?
Yes, I can kill myself.
Have you seen the movie trailer that someone made? It's like the honest movie trailer for Frozen.
The what?
The honest movie trailer for Frozen. You should Google it. We should watch
I
it.
will watch it. Is it safe for my kids or no?
Yay.
I'll watch it and then I'll make sense.
Okay.
And then
I think the only part that isn't is they won't understand it to go over their head anyways.
No.
So
whole time. I
That's
can't.
nice.
Like, what do you mean?
Lectures.
I don't think they'll find it as funny.
So are you coming in at 9 or 11?
Because it's like making fun
I
of
usually
it.
come in at 11. I used to come in at 9. Now
Okay,
I come
the
in at
Histone code. Okay. Okay. So, wow, look at this. That's crazy. I'm going to have to turn the light off for a second. So here is your Histone octamer. Sorry, turn the lights off again. I know it's a no-no for you guys. These are the Histone tails looking off here. Nterminal tails. And researchers have documented what amino acids can get what functional groups put on them. So here, this arginine gets a methionine put on it. This lysine can get a methionine put on it. This lysine can get an acetyl group. Here's a phosphate group put on here. Does anyone know what U stands for? Anyone from cell biology? Ubiquitin. Anyone know what ubiquitin is? Oh, it's mono. Yeah, it's ubiquitin right there. It's one ubiquitin. You're a cheater. Alright, anyway. So, people are unraveling what the code is. It's very, very complex. I'm not going to get into the details on it. The only one I'm going to ask you to remember, the only one you have the opportunity to demonstrate mastery of is acetylation causes DNA to decondense. Acetylation causes DNA to open up because you're getting rid of positive charges and the DNA is less attracted to hang out with proteins, right? Acetylation opens up compacted DNA. Lights are coming on, brace your eyes. The proteins that you could purify and add to DNA in a test tube to cause DNA to open up are called HATs. Histone acetyl transferases. They take acetyl groups and they stick them on histones. That causes the DNA to be less tightly associated with the histones, so the DNA opens up. And you can do this in a test tube. You can purify DNA out of a cell and split it into 3 tubes. You can take the normal untreated cell, untreated stuff. So let's look at the assay for this. And I'm defining for you, when you walk out of this class, you need to know what I mean when I say assay. What's the assay? What's the readout? It's the experiment you do and what you're looking at. What's the assay? You guys get that? So the assay is, I'm going to have 3 conditions. A purified DNA. This is untreated. I can purify DNA. And I'm going to add an histone acetyl transferase to it. And I'll have a tube also from the same prep of DNA, and I'll add a histone deacetyl transferase. HDAC is what it's called. In this case, I should have some compacted parts. Compacted parts. Yeah. And some that are not compacted because I just purified it from a cell and it's a mix and match of it. If I add histone deacetylase, the histone deacetylase should cause the DNA to open up. So in this case, I'll have beads on a string followed by tightly compacted stuff like this in my electron microscope image, right? Followed by areas where the DNA is separated out like that, right? In here, what is the assay? What's the readout of my assay? It's all uncompacted. I won't find any of these densely compacted areas. If I have the deacetylase, things that take acetyl groups off, what is the readout of my assay? All the DNA should be highly compacted. And the beads on the string will look like this. So my assay is whether the DNA is compacted or not. And I do that by looking in an electron microscope. Right? And then you can get fancy and do things like, okay, in this condition, I'm going to take another bit of DNA, all from the same DNA prep, because we want it all controlled, right? I'm going to add an HDAC, and I'm going to look at it, and I'll see that. And then I'm going to take some of it, and afterwards, I'm going to add a hat and incubate that there, and I should be able to show that this all unravels, right? In a test tube, you can do that, and it works. Now, I drew it up here in the space of 2 minutes, and I showed you these experiments. I hope you're learning that research takes a long time. This experiment that I just outlined for you was probably 1, if not 2 PhDs. So we're talking 7 years to do this. 7 years to develop the assay that you can do these readouts. So isn't that crazy? This is why I'm no longer a research scientist. It takes me 2 minutes to think of the experiment. Awesome, and that'll tell me what the answer will be. And 7 years to get the answer. Oh man, I can't do it. For 7, you got two minutes of brilliance, and then no thinking for 7 years. No original thinking, because you already did it in that little 2-minute time span. I was also not very good at it. I had clumsy hands in the lab. That's no good. So HDACs remove these guys so the positive charges remain and the DNA will be packed. All right, that's where we're going to leave it. Hats and HDACs. These are all